Kinases, in simple words, kinases bind a peptide or protein substrate. They're quite varied. And they also bind adenosine triphosphate, which is the biological reagent that gives up a phosphate to the peptide. This molecule has features. It has a large hydrophobic group known as adenine that always on the kinase binds into quite a privileged pocket on the enzyme, on the kinase surface, or sort of into the kinase surface. And what was noted, in fact, Gleevec, the compound I mentioned, is what's called ATP competitive. There's such a privileged pocket on the kinase surface that many small molecules, they go for that pocket. It's like, I found my home. So a lot of the early kinase inhibitors, and even some that are being developed now, are what are called ATP competitive. Now, a fairly large problem with a lot of assays for kinases is that they are run at very low ATP concentration. For example, when you're using radioactive ATP, you can't use the proper physiologic concentrations of ATP. You're much lower in concentration. What does that mean from a practical perspective? It means in a test tube, my inhibitor looks really great because there's not much ATP around. In a cell, my inhibitor doesn't look so good because there's a ton of ATP around, bouncing my inhibitor out of the active site. So there's a bigger and bigger drive to find kinases that adopt different mechanisms. They're a little harder to find. They're called allosteric inhibitors, meaning they bind someplace else, not the active site. They are also called types of inhibitors known as covalent inhibitors that exploit a way of modifying the enzyme more permanently so the ATP or the substrate, the normal substrate, can't bounce the inhibitor out. But in order to analyze these inhibitors, you need to use pretty sophisticated enzyme kinetic approaches. And those approaches truly are provided by this Fluorescence technology. Because of your ability to report activity in real time, you get a much better grasp on how your inhibitors are working and how well they're doing the job that they need to do under physiologic conditions, not under simple clean test tube conditions. Phosphorylation can be viewed as the sort of the light switches in the cell. It's a dynamic process. Nature uses phosphorylation. There are about 520 protein kinases in what's called the kinome. A kinase is the type of enzyme that transfers a phosphate group to a protein. All the proteins within the cell may be in a steady state, but when you want to activate them, there will be an enzyme that, a kinase that will phosphorylate those enzymes and literally turn on an activity. And so all these networks and pathways within cells really are driven by kinase activity. So all the different kinases really regulate the traffic of pathways through the cell. And what's very clear now, and it's actually the target of numerous biotechnology and pharmaceutical companies, is that when kinases go wrong, cells go wrong. So the aberrant behavior of kinases is something that you really want to be able to target as the therapeutic strategies. The first proof of principle happened with Gleevec, that's a well-known kinase inhibitor, and actually proved the principle that if you attenuated kinase activity with a kinase inhibitor, you would attenuate the disease states associated with that kinase. So what my group does is we try to develop sensors to measure the activity of different kinases. And these are sensors that would measure the activity in a test tube if you were trying to develop an inhibitor. They're sensors to measure activity in a cell or tissue lysate if you were trying to understand the profile of a kinase activity in a diseased cell or tissue relative to a healthy tissue. So the technologies that we develop are based on fluorescence. Fluorescence is a fantastic readout to be used in high throughput. It's the emission of light energy. And what my group has done is developed a really clever way to take short peptides that are recognized by kinases, We put into those peptides synthetic chemical components, which are known as fluorophores. When the peptide is phosphorylated by the kinase, the fluorophore lights up. It's a direct read of the kinase doing its work on the peptide. The peptide is a surrogate for a protein substrate. And so this technology, I think, was so exciting when we first realized it. Was almost by accident we realized that it would be so powerful. We were originally developing sensors for different things, for metal ions, and then one day we had this aha moment that said, wait a minute, if we can sense kinases with this method, this is going to be really pretty big. So quite a few years ago I started establishing the intellectual property and gaining patents on the technology. It's gone through 3 generations, each time improving the technology for much more robust use by not just my group or my friends group, but by people at large. And that's the basis for the technology that we're developing in a small company. We also look at other interactions. We develop other probes that are for other critical reactions in the cell. And the second, I would say the second most important interaction is when proteins interact with each other. Not just because of phosphorylation, but just literally there are changes in communication between proteins that trigger pathways. And so we also have fluorescent methods that we can plant into particular partners, one of the 2 partners, that will give a fluorescence response when there's a partnership. And that's another sort of key area where assays can be developed based on fluorophores that we've designed, developed, and really vetted in the group.